Thank you so much for having here. I'm really so grateful for the opportunity to speak, and especially here. This is a very special place to me. I did my doctoral research here at the Miami Project to cure paralysis under the mentorship of Dr. Adele Field-Fote when she was still here. And so it's really so special for me to be speaking here, especially in this room. And so today I'll talk to you a little bit about different things that I did when I was here, actually at the project. And as my current work is focused on understanding the neurobiological underpinnings of this relationship between the practice of regular exercise and the amelioration of cognitive deficits in people who are aging. And so I have a roadmap for you here today. And so we're really going to focus on understanding the link between the practice of regular exercise and the promotion of cognitive brain health and aging. And so I'm going to start talking to you about some of the evidence in humans, and then I'm going to go into the evidence in animals and tell you why, and then go back to the evidence in humans to talk about some of the work that we're doing. We're really excited to be doing this, translating the evidence from animal models into human trials by measuring neuroplasticity changes following exercise. And so I start talking to you about the silver tsunami, right? And so I teach neuroscience for people who are in their doctoral physical therapy program. And so first day of class, I always start by asking people, all right, who here likes geriatrics, likes working with older adults? And really a couple of hands go up. And then I talk to them about the demographic patterns, right, of our country and our world. And many of them are not at that moment in the first sort of day of class aware that soon, very soon in 2030, 1 out of 5 people will be 65 and older. And then in 2050, that's already coming as well. 1 out of 4 people will be 65 and older. And so I always tell, so you like sports? You're going to like sports. I need to talk closer to the microphone. If you like sports, you're going to love sports geriatric. If you like women's health, you're going to love, working with older adults. And so this is one of the reasons why we need to understand what things are important for these people, because they are the fastest demographic currently growing in the world. And so on the right side here, I have a graph for you. That's just one of many. You can many. Am I doing this wrong? Yes, Iggy, sorry, I had it backwards. And so if you ask people who are aging, what are their priorities with regards to their health, they overwhelmingly will say that being able to the maintenance of a sharp mind is really important to them. And this is not surprising, because this is highly tied to their ability to continue living functional lives, independent lives, right? And so here, we are really familiar with the concept of hurricanes, right? And so there are some interesting analogies there. And so unlike hurricanes, age-related cognitive decline is really difficult to forecast. And so when a hurricane is coming, we sort of know, and so we can prepare, and so that's really an important thing. But with age-related cognitive decline, we don't have so much of a warning, okay? And so, but akin to hurricanes, the impact can be quite devastating for the individual, for their families, and for the society at large. And so in 2015, there were about 47 million people living with dementia worldwide, and the global cost in 2015 was upwards of $800 billion. And so the projection is that in 2050, again, that's coming quickly, this is going to grow to 131 million people around the world. Right? And so this here justifies and makes a really compelling case for the unmet need that exists for effective, for accessible therapies to mitigate age-related cognitive decline. And so I have a picture here on the, if I can work this, I have a picture here on the right side, and this is one of the situations when the statistic touches your life, right? And so over here, I have my grandfather, right over here. Dionisio is his name, and was his name, and he passed away from complications related with Alzheimer's disease, and it was a really hard time. About 15 years of really hard time for our family. 8 years of this he was bedridden, and this is not uncommon. And so, just if I may ask, in a show of hands, has this statistic touched any of your family or of your lives? Right? And so this is something that it's really, really important for us. And so and this is one of the reasons why, before I was here and I was talking about interesting things, again, with brain stimulation and focused on spinal cord injury, but as this statistic touched my life, the science became so much more compelling and interesting to me to tackle. And so it's a complicated problem. I'm not going to go over this slide. The only thing reason why I'm mentioning this slide here, on the left, you have cognitive trajectories. They're extremely variable in people, and this is one of the difficulties, right, to make this case of why this is so difficult for us to tackle. And so here, these things that to me look like a feather, right? It sort of looks like feathers. These are the cognitive trajectories of over 1,000 older adults between the ages of 70 to 76. And the reason why it looks like a feather is because one goes up, one goes down, one goes up, one goes down, one goes up, one goes down. And so these are, speed, verbal memory, different constructs of cognition. And so the trajectory is quite variable, which makes the ability to predict very challenging. And this creates a problem, like clinically. If somebody comes to you and they start to complain about subjective memory complaints, you don't know if they're on a path that goes like this or if in a path that goes more like this, right? On the right side here of this graph, I have just another point to show you. So this is quite interesting. They were actually characterizing people on the left who had high cognitive performance, and on the right, low cognitive performance. And this is looking at the neuropathology here. And you can see here that even for folks who have high cognitive performance, they can sustain a quite high burden of neuropathology that is indicative of Alzheimer's disease, vascular injury, and Lewy body dementia, which is something that is to just make you aware of the fact that we do have diagnostics and they have proven to be somewhat useful, but they're not perfect, right? Otherwise, this wouldn't happen. But it also presents a really interesting opportunity, because if you have these folks here, right, that have high cognitive performance, but their neuropathology resembles that of dementia, that means that there's something that they're doing that is preserving their cognition that we need to understand better. And this is a really, really exciting thing. So there are many things about your golden years, right? When you reach your golden years, there are many things about it that you can't change. You can't change your prior experiences. You can't change, the level of neuroinflammation that you have, and some of the things are related to who you are as an individual, to your outlook in life, to your genetic makeup, and all of those things. But there are some opportunities, and so the current evidence does indicate that by changing your physical activity, by decreasing your sedentary behaviors, you can take steps to steer a little bit your golden years into a better stage. And so this idea, right, that a healthy mind lives in a healthy body is not new at all, right? This is something that we knew when we were back in the Roman times in the 2nd century, depending on how you count your centuries. But essentially now is a really exciting time because powerful organizations, right, such as the American College of Sports Medicine and the Institute of Medicine and the Academy of Science, Engineering, and Medicine and the U.S. Department of Health, they have all together agreed that engagement in physical exercise can be helpful, to promote cognitive brain health. And I left here the sort of little logo from the ACSM saying that exercise is medicine, because I wanted for us to think together about that, right? Exercise is medicine, and that's really great, but if exercise is indeed medicine, then we need to be able to have the ability to prescribe exercise with the specificity that other types of medicine are prescribed, right, like pharmacotherapy. And so this is the same point that Dr. Gator was making last week when he talked. And so what we want to know, right? And so great. So let me think back to my grandfather, right? And so exercise, all of these, the U.S. Department of Health, everybody says that exercise contributes to cognitive brain health. You should be exercising. But that's really great. But when you're a clinician, right, what do you tell your loved one? What do you tell your patient? What could I have told my grandpa? Is it that he needed to run every day, 5 days a week? Or is it that he needed to is walking okay? would he need to do that 5 days a week to get an effect? Or if he just does it twice a week, is that okay? Because he had bad knees when he was able to walk, right? And so what's the session duration that's sufficient for optimal gains? What's the sessions per week and total weeks, right, for him to be able to reap those benefits? And if he did do that, would his memory improve because he was having trouble with that, or would other things, his concentration or his executive functioning or other parts of the cognition, right? And do you have to be running all the time? Because we see this all the time, the New York Times, right? They say, oh, running is really good. Running, you should run. But what if you can't run? Is walking okay? Is dancing okay? Is swimming okay? All of these things we need to know because essentially people, it's hard to get people to exercise, and they will do what they like to do. And so where does the current evidence stand on the optimal dose of exercise to promote cognitive brain health in older adults? And so we took a little journey to be able to find this. I was really surprised because coming from spinal cord injury research, I think we have a lot more delineated in terms of optimal activity and therapies and rehabilitation. And so in this it's really interesting sometimes to go outside of your immediate field because you can often be surprised. And so when I came in to look at this in aging, we really had nothing. And so we had the evidence. There were over 50 systematic reviews that went and said exercise is good for the brain in aging. But that says nothing about the dose. That says nothing about how I can prescribe people, because prescription is important too. For example, right? Exercise is good to help people lose weight. But if you prescribe exercise for somebody and it's been a month that they're engaged and they haven't really met their goal, you can go back and say, no, it's okay. It's not that exercise is ineffective, it's that you need to go for longer. And so having this information about dose is important even to get the buy-in from our patients. And as you know, in the healthcare community, we need to do a lot better in really motivating people to change their behavior, but that's a separate story. And I'll go back to this. And so we did this quantitative systematic review, and we're really excited. It was fun. We got a lot of interest from the media, and it was one of those fun things that happen sometimes when you do these kinds of things. But essentially, we went back to the body of literature. So for many, many years, right, we've done, right, all of these meta-analyses that gave us effect sizes saying that exercise is good. Now, this thing, what we were doing was we went in there to say, okay, exercise is good. How much exercise was given to be good? And so we wanted to get all of the randomized controlled trials that we could find that proposed exercise and had a neurocognitive assessment. And we wanted to quantify the dose and run statistical tests to see if we could get some part of the dose to be responsible for the gains. Furthermore, neuropsychological assessment, also unlike, one of the interesting things about changing a little bit of your shift is that there are so many different outcome measures. And so a lot of them overlap. And then you get an article that says exercise is good, and this was the test that was used that measures memory. And then you get another that says exercise is good, and there was this other test that was used to measure memory. And then you get another one that says exercise was bad, and we use this other one for memory. So what is the thing, right? So we also wanted to measure the consistency of effects. If from all of the studies in memory, half of them said that exercise was good and half of them said that exercise was bad, that's not very consistent, right? And so we did that. We did some assessment of the quality of the articles, and these were about 100 randomized clinical trials that yielded data from about 11,000 people. These were all interventional studies. Okay, and so I won't go into much detail about this because I have more stuff that I want to share with you. But what I will say is that when we looked at all of these things, session duration in minutes, length of the intervention in total weeks, length of the intervention in total hours, length of the intervention in minutes per week, sort of like how the American Heart Association does, frequency of visits per week. So the only aspect of dose that was a significant correlate of improved cognition was actually the length of intervention in total hours. And so when we went in and we dug in a little deeper, we saw that the studies that had a median amount of total exercise that was distributed, in many sessions over many weeks and stuff. The ones that had a median of about 50 hours, 52 hours, they were more likely to show improvements in exercise compared to the one that had median of 30-something hours. And so the interpretation of this was actually pretty encouraging for us, because it doesn't seem to matter if you are able to do the exercise for 30 minutes a day or 45 minutes a day, if you can fit it in twice a week or 5 times a week, but the cumulative exposure to it, once you do what you can do, and you do that for about the accumulated time to get to your 50-something hours, that's when you can get something back. You can reap those benefits, right? And so I think about it in chips. Like, I put my little, chip, I did this amount of exercise, and once I rack it up and I've got 52 hours, this is when something's gonna happen that's good. And this is good because if you have your grandpa or you have your dad, like I do right now, and you're encouraging him, you can say, hey, Dad, how are you doing? No, you gotta go longer. You gotta go longer. You gotta get to that 50-hour mark. It's not that it's ineffective, it's just that the dose wasn't sufficient. And so in other things, it seems like aerobic exercise was the most often used mode of exercise, right? But it wasn't the only one. Resistance training was there, right? And so all of these things are backed up by the evidence, and a combination of them. And also we were really excited to see that mind-body exercises were also represented there. And clinically, this is important because some people are recovering from injuries, some people have bad knees, and so being able to do something that's a little bit of a lighter intensity is also important. And it's important to know that it can contribute to your cognitive brain health. And so with regards to the consistency of effects, right, it seems like from all this body of evidence, the most consistent effects that we saw were in the ability of the brain to process information, the processing speed and attention, and also executive function. And this is another super encouraging finding, because these two constructs are among the first that start to deteriorate in aging. And so this is actually evidence that you can show to your loved ones that you can turn back the clock of aging by practicing physical exercise. And so this is another little interesting thing that I'll talk to you about as well. And so just for us to think and dream, right? So how much would dementia risk change if we eliminated modifiable risk factors, right? This was a study, cool study for you to read if you're interested in this kind of thing, commissioned by the Lancet. And so the population attributable fraction calculates the percent reduction in new cases if a particular of the risk factors was to be eliminated, okay? And so with this, and of course there's, and you'll see, and there's limitations like there is with any study, but it was interesting to see that their calculations was that about 35% of the risk of dementia is attributed to potentially modifiable, right? And you have here, this is kind of hard for you to see, but over here in the blue on the top, you have things like, this is supposed to represent the lifespan. And so you have here your APOE allele, right? And you have here education, and you have here hearing loss, and I see Hillary Snap here, and hearing loss is a really important one because it's very prevalent. But you have your hypertension and obesity in midlife, and in late life you have here smoking, and you have depression. When you look at physical inactivity alone, right, if we eliminated the risk due to physical inactivity, we would decrease by 3%. But if you look at collectively, right, the associated conditions, right, because physical exercise is going to potentially impact things like social isolation, like diabetes, like depression, like hypertension and obesity. The number is much higher. It goes up to 13%. So this is a really great opportunity for us to tackle. So in my lab, we have a dream, and this dream is the long-term goal, to develop these individualized exercise interventions to promote cognitive brain health in aging adults. And so we want to find out what's the best dose, not just for any person, but for the person, for the individual. And so when we think about that, and I have Dr. Nash here in the room, right? Exercise works for everything, and maybe this is one of the things that he'll tell me when I'm done. But when I think about exercise working, let me think about exercise works for strength. I know that. Exercise works really well to improve strength. Before my PhD, I did physical therapy, so I know this stuff. And it works to improve strength because we have a way to individualize the exercise prescription for these two very different classes of individuals. You wouldn't give the same amount of exercise, the same mode of exercise, the same, periodization of exercise to these individuals. So why would you tell all older adults to just walk, right? We need a sensitive measure to capture the changes in the brain following exercise in order to be able to prescribe exercise with the specificity that I believe we can do. And so then we go back, right? We're getting now into the animal part of thing, the second thing that I told you about. So what are the active ingredients? This is a really complicated one because exercise does everything. And so at a cellular level, right, you have exercise, it increases cerebral blood flow, it improves endothelial health. Endothelial health is really, really important. And it decreases oxidative stress, and it decreases neuroinflammation, and it also is going to promote the transcription of growth factors that are really, really important. So brain-derived neurotrophin factor, VEGF also, these things are really important for the cognitive gains that follow exercise. I'm not going to go into great detail in this, but just to show you how complex this figure is. And then when I go from micro to macro, there are things that are helping, right? Exercise helps to preserve real estate, right? So decrease atrophy in the brain, gray matter, white matter, glucose metabolism. It also increases hippocampal neurogenesis, which is something that's really important and tied to our ability to continue performing having high cognitive performance as we age. And so in this scenario, I want to argue with you guys that the functional impact of all of these changes that are happening are going to culminate, let's say neurogenesis, for example, right? This is going to culminate in the capacity of this new neuron or this enhanced metabolism that reaches this new neuron to incorporate itself into the existing functional connections, right? Display action potential and synaptic activity. And so I don't want to say things like a common final pathway, but almost, right? The way that you would assess, the way that the link between these molecular changes and the actual behavior is synaptic activity, right? And synaptic activity is assessed with electrophysiology here. And so I have here a picture of long-term potentiation. I'm sure many, if not all of you guys are familiar with this. And so here you have, this is done in hippocampal neurons and in animal models. And so after here signifies synaptic activities, right? So excitatory postsynaptic potentials. And so after a brief period of high frequency stimulation, you can see that in comparison with the baseline synaptic activity, there's a potentiation of the synaptic activity that lasts for about an hour after you do the test, right? And so this is, the neurons that fire together, wire together. Long-term potentiation is really important, not only because when you go back to the animal models and you see the link between exercise and cognition, it's necessary for long-term potentiation to occur. And so one of the main mechanisms by which neurons that are generated incorporate themselves into the circuitry that's going to produce cognition is because there's an enhancement in neuroplasticity. And so this is one of many examples. You have here again baseline synaptic activity. At the point of 0 here, you give this high frequency stim, and then you have an elevation of the responses, and this is the potentiation, post-tetanic potentiation or long-term potentiation. In this study here, you have rodents that were given a running wheel or not given a running wheel. And you don't really need fancy statistics to show that these two groups are different, right? In this study, and it was one of the really important studies, they were able to show that the enhancement in neuroplasticity was correlated with improved cognitive performance, visual spatial performance in the animals, right? And so linking the improved neuroplasticity to the improved cognition. Now let me talk a little bit about this with you. One of the things that I, in looking at this literature that I see that is somewhat of, that doesn't it's not super productive is that we often generalize too much. And so we might do a study in humans, and we might show that the study shows improved cognition in following exercise, and then we go back to see what would be the things that would be responsible for it, and we cite a study in animals that did exercise for a much brief period. And we say, well, in that animal study, it was shown that there was increased neurogenesis, and so that's maybe why this is happening in the humans that I am exercising for a month, right? And so we lose a lot by doing that, and this is one example that I'll give you. So this is a study in animals, right? And it was a study that measured neurogenesis, hippocampal neurogenesis, following exercise, and it also measured synaptic plasticity. So here you see that with 3 days of running, there was increased neurogenesis. With 7 days of running, there was increased neurogenesis. Something happened with the 14 days, and they don't know quite well why. But in the 28 days, there was increased neurogenesis as well, right? And so if you were only to do this part of the study, and maybe you would see that one of the implications of this new neurogenesis is that the animals would have greater neuroplasticity, greater cognitive performance. But let me show you just what happened with the plasticity. So I've got here the same sort of graphs, right? And so with 3 days of running, neuroplasticity didn't improve yet. With 7 days of running, with 14 days of running, with 28 days of running, and then they kept going. But they saw that with 56 days of running, then you saw the increases in neuroplasticity. And so sometimes, right, it's really important for me to make the point that mechanisms are dose-dependent as well. And so this was another study that we did this year where we were just reviewing the literature to try to reconcile some of these discrepancies. And we had a simple idea. Well, let's look at the mechanisms that are believed to underlie the improvements in cognition. And let's see really when they happen with respect to dose. And what we found was really interesting here. And so we have all of these things that are hemodynamic or connectivity changes or structural changes or trophic factors. And we have the different types of modes of exercise. So what we find here from 1 day, 10 weeks, 12 weeks, up to 2 years of exercise is that cerebral perfusion changes and connectivity changes happen at the short term, 4 months. When you get to around 6 months, you start to get these structural changes that we talked about, such as the gray matter and the white matter. But once you get to about 1 to 2 years, you have more consistent improvements in structural changes. Now, one thing that I want to tell you here is that we limited here in this graph the human data. So we weren't really able to see when the plasticity changes because this is done in animals. But I'll show you some human stuff, and this is where we get back to the human. So the knowledge gap here is that the classic measures of synaptic neuroplasticity are measured with electrophysiology, and we are really excited to be among the first people that apply a measure that was translated from this long-term potentiation paradigms into humans who are exercising in the context of aging. And so this will allow us to probe the circuits that are responsible for synaptic plasticity and introduce a new way to potentially assess dose response and propose more individual prescriptions. And so this is, it's done with transcranial magnetic stimulation. And so this is the long-term potentiation-like test that can be done in humans. And so essentially you have here motor evoked potentials, and so these are recorded from the motor cortex with surface electrodes. And so you're able to elicit a high-frequency stimulus akin to what's done in the animals, but in the human with induced current with the transcranial magnetic stim. And then you measure the amplitude of the motor evoked potentials after the stimulation, similar to what you do in the synaptic activity. And so what you see here, and so I want you to focus on the blue part of this graph, is that following that high frequency stim, you have a period of enhanced amplitude of motor evoked potentials that shares many similarities with the with the classical experiments of long-term potentiation. So one thing that's similar between them is the time course of potentiation is similar. Another thing that's similar is the biochemistry of it, and so they're both governed by the same biochemical processes. And so essentially you can have an assessment of neuroplasticity in the human by doing this. And so this is a relatively new paradigm with TMS. And so, but there's some pilot data to show that just like cognition decreases, right, in aging, this measure of neuroplasticity was also shown to decrease in people who are aging. Furthermore, there was another study here that showed essentially that their impairment in this plasticity response to this test that were present in people who had type 2 diabetes, who had cognitive deficits. And so the extent of the impairment in the plasticity response was correlated with the extent of impairment in cognitive testing. And so one of the things that you might tell me is that, well, but you were, in the animal studies, you were recording and stimulating the hippocampus, but now you're talking to me about the motor cortex. Yes, that's a limitation, just like any, neuroimaging or any, technique that we use has limitations. One of the limitations of this technique that we're using is that we're getting insights into the motor cortex, and we're doing so because we have an input and an output. But we are addressing this limitation by combining the TMS with EEG. And so I'm really, really fortunate to be an awardee of the Clinical and Translational Science Institute KL2 Award. And so for the junior people in the room, the CTSI is a wonderful place. And it's really made a difference for me. And so by doing this, I essentially have this grant where I have this wonderful team of mentors who are going to help to navigate this project that I'm going to propose and talk to you about. And so there's two years of protected time. I have 75% of my time dedicated to seeing this project through, and I have the help of the mentors. It's really hard to navigate the career and the scientific path when you're a young, early career investigator. So this is a really, really wonderful mechanism. And so I have a wonderful, distinguished group of mentors. I have Dr. Tatiana Rundek here in neurology, Dr. David Lowenstein at the CNSA, the former Center on Aging. I have Alvaro Pascual Leone at Harvard, who's my brain stimulation mentor. I have Dr. Lawrence Cahalan, who's a consultant on my, with regards to the exercise testing. And I have also Marcela Kitay-Gorossi, who works with Dr. Lowenstein and is helping me. And so essentially, the second part of the talk, I'm going to talk to you about this, my K project, where we are essentially putting these ideas to the test. We are recruiting aging adults from the community, and many of them from the University of Miami community, and we're exercising with them for 2 months. Before and after we do that, we are collecting brain neuroplasticity tests. We're collecting a comprehensive neuropsychological battery. We're also collecting some data on their cardiovascular fitness because, as you know, one of the things that I didn't even have the opportunity to talk because of the time is all of the effects of cardiovascular fitness. Cardiovascular risk factors happen to be cognitive aging risk factors. And so people are going to be exercising. They may be modifying those risks and thereby improving their cognition. So we're taking a look at that. We're also going to look at some genetic stuff that I'll tell you in a bit as well. And so with regards to the genetic factors, we're looking at effect modification. And so obviously the long-term goal is to be able to individualize the prescription. And one of the ways that one might do that is looking at the genetics. And so brain-derived neurotrophin factor is one of the main, most consistent increases in trophic factors that are correlated with the improvement in cognitive gains following exercise. And so it's important for us to measure BDNF levels. But furthermore, in about 30% of the population, there's a single nucleotide polymorphism in the BDNF gene that essentially leads to a decreased activity-dependent release of BDNF. And so this would, in theory, render some people plasticity deficient. And so it's interesting for us to be able to understand if these people might have, does that influence their potential for their gains in cognition through exercise? We don't know. We're going to take a look. In addition to that, the APOE4 allele, right? And so that's probably floating around my head as I have this family history, right, of AD. But one of the interesting things that seems to the preliminary evidence is showing is that even though it increases your likelihood for developing dementia, it actually may have people with an E4 allele may have an increased potential for gains with exercise and aging, which is really interesting. The mechanisms are not very fully known, and so we're investigating this as well. And so we have a lot of neuropsychological tests. One of the things that we really wanted to do was to have a more comprehensive battery. This is also one of the limitations when I went to do that big review as well. Some of the tests that, you talk about things like executive function and memory. Some of the tests that people are using are cognitive screening tests, like a mini-mental or a MoCA. And so it's important for us to have more detail and overlapping tests and be able to really, really, look at people and look at how they are, within their age and their education level, and have a very careful look at their cognition. And so we're excited to do this. So we have many, many constructs of cognition. We're sort of zeroing in more on that executive function and attention because this is what the evidence shows that is most likely to improve with exercise. And so our inclusion criteria, this is a moment for my plug here. If you know anybody within the university community, or, we've got friends and family of university community, people who are not associated with the university at all, we're looking for people who are 55 and older, who have no clinically detectable cognitive impairment. We'll screen them well, who have low activity level. Primary English language could be English and Spanish, and who don't have any unstable medical conditions that would render them not able to exercise or not able to undergo the TMS test. I'm not going to talk about that, but we screen them really well. We have to take a look at their medications. I won't even go into much detail. So let me talk to you a little bit about what we have, right? And so we started in probably March or April to enroll these people. And so we yesterday enrolled number 25. We finished number 8. And so I'll show you a little bit of data from number 8. We hope to go to 80 by December of next year, but it's a little bit ambitious. And so right now we have a gender balance sample. We have many Hispanics, and that's really exciting because Hispanics are often underrepresented in clinical research trials, especially with some of the things that are happening now in the country. And so it's important to be able to have a chance to characterize them. They're mostly well educated, and most of them are active in the workforce, many at UM. And so we contacted about 124 people, and we have just a breakdown here of how this goes. Trying to get people to come and exercise for 2 months with us is a little bit of a hard task, but we try to be to do the best we can. And so I have here some very, very preliminary graphs to take a look at the brain plasticity. But I'm gonna back up a little bit, because I think I wanted to make just one thing that'll be more clear. Let me see which graph is best for this. I'm going to go here. All right, so essentially the test consists of coming in, right? So we measure the motor evoked potentials at baseline. We then give this high-frequency stim that opens up this window of enhanced plasticity. And then we come in and we do the same thing to measure it again and see what did this high-frequency stimulation do? What was the overall plasticity, right? And so essentially, 5 minutes after you gave that high-frequency stim, you start to get this increase in the amplitude of the response. And so the height of this is essentially the measure of plasticity. So the higher it goes, the more plastic, okay? And so the most consistent points have shown to be the earlier minutes, like 5 and 10 minutes after the stim is given. And then overall, over the course of 45 minutes to an hour, the activity comes back to baseline, which is good. And we measure that and we send people home, okay? So essentially, the height of the curve at these earlier minutes is the indication of plasticity. The higher, the more plastic, okay? So we have really nothing to show you in terms of, we wouldn't look at anything really with 8 people, but I just wanted to give you a flavor. And so here I have, right, 5 minutes after we gave the stim, and 10 minutes after, and 20 minutes after, and 30 minutes after. And so things are all over the place. There's a lot of variability, and this is not uncommon in electrophysiological studies. But one of the things that we do notice is that a lot of the responses at pre-intervention are sort of lower than the 0%, right? So they're not really potentiating. It's like they're showing an impaired response to the test. And so with these 8 people, it seems like we do get a little bit more of a lifting of that curve. You can see in the red the mean. And so with just this few number of people, it seems like the plasticity is increasing. But, we'll see this through with 80 people and see what happens. I've plotted it here in another just to show you the mean here, right? From having a negative response to having a small positive response. Again, by itself, this really means nothing. With 8 people, it means nothing, but it will mean something when we look at the cognition, when we look at the genetics, when we look at the cardiovascular fitness. And so we'll be able to have a really interesting thing to talk about. And so here we're looking at the R bands. It's essentially a big test that assesses multiple constructs, and so immediate memory, attention, visual, spatial. And so just with these people, even though there's, again, much variability, there seems to be some, early patterns of improved attention and visual spatial performance, which is consistent with the data. Same thing happening here with executive function and processing speed, the Stroop, and the Trails B. Trails A is more for processing speed. But another thing that we're really excited about is that we're using this cognitive stress test, which is a paradigm that was developed by David Lowenstein, doctor here at the CNSA. And so this is really, really interesting. It's essentially a list learning, but one of the things that Dr. Lowenstein has shown is that there are specific abilities that seem to be really, really sensitive to changes in aging. And one of them has to do with these sort of list learning. And so in this cognitive test, like many of the cognitive tests that we do, we give people a list and we ask them to recall, right? And then we give people another list and then we ask them to recall that. But there's something that's called this proactive semantic interference that essentially means that old learning interferes with the new learning. And so when you give the second list again, you see their ability to recover from that interference. And this specific thing is really, really, really sensitive to preclinical cognitive decline and also amyloid, right? And so one of the neurobiological markers of age-related cognitive decline. And so we're really excited to be using tests that seem to be more sensitive so that we can have further insights. Other things that we're really looking and excited to look at is things like self-efficacy related with exercise. And so there's a lot of things here. It's super busy, but this is just for you to see an exercise self-efficacy test. And essentially, you're asking people if they're very sure, pretty sure, a little sure, or not sure at all that they're going to do exercise in all of these scenarios. My personal favorite is when there are other more interesting things to do. And so you can see here that red is bad, right? They're not going to do it. But you can see in this sample that we have that we're seeing more colors. And so this is exciting to be able to, just with 2 months, to get people who are couch potato and older to change their self-efficacy with exercise is super exciting. Another thing here has to do with their barriers, right? And so again, like, what are the barriers to exercise? I'm not interested in physical activity, right? And so do you agree, partially agree, or disagree? And so in this case, red is good, and we see more red at a post. And so these are the things that perhaps are less mechanistic, but they're really important and clinically meaningful. And so the next step is that I'm actively preparing a grant to go in February. We'll see which one. It seems like it'll be still an NIHK. It'll be just continued career development, but we'll see. Maybe the data is incredible and we'll go for something more ambitious. But we really want to figure out ways to more, in a more operational manner, individualize exercise based on things like, cardiovascular fitness and genetics and plasticity, and see if we can improve the effect sizes, which are present, support exercise, but are a little low, right? And so comparing different types of exercises and dosages, I didn't tell you that we're just doing aerobic exercise with these people, not because I think it's best, it's just because you have to start with something simple. Aerobic exercise is really easy for us to know physiologically what's happening, but all of the things under the sun are important. I wanted to thank so many people, some of them who are here, people in all sorts of places, primarily my dream team, Jordan Rice, Danilo Cabral. They're downstairs doing a plasticity test right now, so they can't be here. We are on the first floor in the hallway before the laboratory of Dr. Mark Nash. Christina Nunez, Kaylee Van Dusen, Danielle Abel, Babak Mustafi, Dina Cillian. And I also wanted to, for any young parents in the room, or maybe you will be, I wanted to just tell you just a life thing. The day that I was awarded this grant, I had a baby who was 8 weeks old, and now I have a baby who is like that. I wanted to acknowledge the village around me. It's really hard to be a woman, to have a baby and to do science, and I have many people to thank for that. The person that I'll thank right now is Marilyn Cárdenas, and she allows me to have the seemingly impossible number of full-time jobs that are to be the mother of a little one and do science and teach and mentor and do all sorts of things. And so with that, I hope that you have questions and things for me to include in my grant in February. Thank you.
So exercise is a mix of sensory stimulation and motor output, preparation of motor programs and everything else. And it's, if I recall correctly, just even stimulation of pathways can increase BDNF factors in the postsynaptic neurons and things like that. So there's a lot of things going on. But have you thought about, if you want, the question is, why is this working on cognitive things? And it gets input and has output also that relates to sensory processing and motor output. But have you thought about trying to do more to maybe even potentiate the effects of the exercise, for example, by doing some kind of cognitive tasks? So reading, reciting, any kind of thing that could maybe engage after exercise, the cognitive area.
And I was instructed by Iggy to repeat because of the video recording. And so the question from Dr. Noga would be that there are many things acting in exercise and input and output, and perhaps considering the relevance of sort of priming the system during or after, you mean, Dr. Noga? After the exercise to maximize, because things like BDNF are going to be released with cognitive activity, with much activity really. I haven't really, but I haven't yet, and I would love to further discuss this with you about ideas, and probably one of the reasons would be. One of the things that I would be interested in discussing with you is salience. And so to be able to match the right stimulus for every person, given that people are so different, and one of them may be that they want to watch Netflix while they're doing something, or one of them might want to hear music, or might want to, from a grant reviewer perspective, does that make the dose of what we're giving different? Okay. Well, thank you very much. Oh, yes?
I have like a couple of questions. More because it's a very exciting talk. Thanks for that. Two questions. One was sort of adding upon, like, what Dr. Noga just said. So there was just a recent article from Stanford that came out that talked about chess players who burn like up to 6,000 calories, right, per day. So that's like a very high amount of activity. Much of that is because of like mental focus that they're doing. So it would be interesting to look at cognitive tasks that they do in addition to just exercise and seeing whether there is a combination effect that you're getting, right? Some kind of this potentiation that lasts beyond maybe 30 minutes, maybe, right? Maybe for an hour, 2 hours, something like that.
And this related with the assessment or related with the intervention? So would you want to potentiate, do you think that perhaps we would get higher potentiation?
That's what I would
wow,
expect,
during
but I don't
the
know.
assessment.
Yeah,
okay,
during the
that's
assessment,
really
but
interesting.
I don't know.
That's really interesting.
Might be one way to do that. And then the second thing was, like, I saw that in the battery of the test that you do, a lot of that is related to cognition, which is what you want to do. But we also know that, hearing and balance, and you kind of alluded to that point, plays a big role. And one of the pathways that it affects is essentially the sympathetic response, right? So it reduces, like, your cardiac output as we age, and there's a balance component to it. I wonder if you've thought about studying that, perhaps.
Yes,
in these group of patients.
I would love to also, I sure would follow up the discussion with you regarding that, but I didn't have time, just because I wanted to make sure that all of you have time to eat lunch. But in this study, we're also assessing timed up and go with a dual task component to have insights into cognitive motor interference, because essentially one of the things that coming from rehabilitation into this that I see is that some of these cognition is very, paper, pencil, numbers. And so cognition is a fluid thing that interferes with movement. And so in my mind, things like your ability to carry out a mental task while you're walking is cognition in the real world. And in my mind, the plasticity test that we do is a measure of how adaptable the nervous system is. And that dual task ability is a measure of how adaptable your nervous system is. And so we're going to look to put these things together. So in all of these people, we have cognitive motor interference. Not so much hearing, and maybe we'll talk about with Dr. Snap, but to see if we can incorporate something. But yes, thank you.
Joyce, how intense is the exercise that you're planning for the paper?
Oh, it's not so intense. It's the first 4 weeks. It's what the IRB let me do, Dr. Nash. Like 55 to 64% of age predicted max in the first 4 weeks, and then 70 to 90 in the last 4 weeks. And so this was, and I would love to hear your thoughts regarding that. We just wanted to do something that was, moderate to high, and just see, because we had a pilot study where we did 4 weeks of moderate, but moderate really seemed to be more light. In practical outcomes, people were more walking, and even from that perspective, for them to get out of their house, they want to be working hard.
And this is all treadmill?
This is all treadmill currently, not because of our interests, but because of, you got to start simple. These neuroplasticity measures have never been characterized with, we were the only people that did studies, but in middle-aged adults with exercise like this. And so we needed to start small and simple. I would love to follow up with you with ideas for the future as well. Well, thank you very much. Please, let me know anything if you're interested in this, and thank you.